Hey, Mr. P here. In this video, we're going to talk specifically about lipids. So in the last video, we talked about carbohydrates. We talked about how carbohydrates are made. We talked about how they're structured. We talked about what the purpose is. We talked about what their structural and functional applications are. And now we're going to talk about lipids much in the same way. So lipids are the second class of organic molecules that, if you remember from our list of four, were outlined. Lipids will be second. Proteins will be third, nucleic acids will be fourth, and so let's dive in and take a look at lipids. Lipids are a large and very diverse group of organic molecules as well. They are generally not soluble in water, and they are made mostly of carbon, hydrogen, and oxygen atoms, but carbon and hydrogen makes up the bulk of the lipid, and most of the time the fatty acid. Triglycerides are one of the big groups of lipids that constitutes the fats and oils. Fats obviously being animal products, oils being plant products. Waxes are a type of lipids, cholesterol is a type of lipid, steroids are a type of lipid, and phospholipids obviously, because it has lipids in the name, is a type of lipids. So we're going to differentiate between all of these types. We're going to talk about what they're used for and really talk about what the structure and function is of these molecules. So over here you'll see that this is obviously chunks of animal fat that is lipid-based. These are your plant-based oils. These are also lipid-based. Both of these, the animal and plant oils and fats, are going to be your triglycerides, which is kind of one of the big groups of phospholipids, or one of the big groups of lipids we're going to talk about. Phospholipids obviously being the other big group we're going to focus on. We're really not going to talk too much about cholesterol, steroids, or waxes in this class, but I wanted to put them on here because they are lipids. But for the most part, it's going to be these two and the phospholipid. This is showing you the chemical structure of cholesterol, which is an important molecule within the body, but obviously not going to talk about it. It's kind of one of the details we're going to minimize. Okay, so lipids, and I'm speaking on the fats and oils, triglycerides, are going to provide plants and animals with long-term energy storage, which is different than the carbohydrates. Remember, carbohydrates were short-term energy storage, whereas the lipids, specifically triglycerides, are going to be long-term energy storage. I think you guys understand that. I think you know that. If you talked about or with anybody about how animals store their energy long term, hopefully fat storage or fat tissue, fat molecules would come to mind. Animals, whether it be wild animals or humans, are going to store their energy long term as fat. Plants, on the other hand, don't get fat. Plants don't store their food or their energy as fat. They store their energy as oils, which is why if you go to the grocery store and look at the oil section, you will only find plant-based oils. You won't find chicken oil or beef oil or turkey oil. That just kind of sounds silly to say, right? It's all of your sunflower seed oil or your canola oil or olive oil or vegetable oil, corn oil. Those are all plant-based because oils are derived from plants. Phospholipids are the other group we're going to talk about. And they are really important because they form biological membranes and they provide waterproof coverings for different cellular components or molecules or structures within an organism. So what is a triglyceride? Triglyceride is a molecule that stores energy long term. It has 3 fatty acids, which is why it has a prefix tri. Okay, 3 fatty acid chains. It has a glycerol head, and they are utilized in both plant and animals. These are your fats and oils, and are produced much in the same way that carbohydrates were produced. Remember, we talked about anabolism, we talked about catabolism, we talked about the building up and the breaking down of molecules. Okay, if you need a refresher, go back and watch. But much like the monosaccharides being linked by dehydration synthesis, the glycerol is linked to the fatty acids using the same exact process. So this is an anabolic process. Okay, and it's called dehydration synthesis, which I know if you watched the last video is not the first time you're hearing of this. Dehydration synthesis is the removal of water to produce a covalent bond to link monomers together. In this case, glycerol And the fatty acid chains are the monomers. Those are the single subunits that get linked together to form a bigger molecule. The glycerol has these 3 hydrogens that are going to be stripped off. The fatty acid chains have an hydroxide group at the end of each of the fatty acid that will also be stripped off. And if you strip off an OH hydroxide and a hydrogen, you're going to get 3 molecules of H2O in this case, which you can see over here as a byproduct. When you strip these individual components off to produce the water, you're going to have to combine this bond with this bond, which is how this glycerol head gets combined to these 3 fatty acid chains. You can see that here. These are the bonds that are produced by removing the water, and you end up with a triglyceride, which is a glycerol head, 3 fatty acid chains, and your end product is the 3 water molecules, which are just a byproduct. Okay, that is dehydration synthesis. It's an anabolic process. It is building larger molecules from smaller ones. Okay, so these triglycerides are going to provide plants and animals with long-term energy storage. That is key. That is the main function of fats and oils, which are the triglycerides. Now, you probably have learned about saturated fatty acids. You've probably learned about unsaturated fatty acids. You've probably even heard of trans fats, right? When you look at nutrition labels, you see saturated fats, you see unsaturated fats, and you see trans fats. For the purposes of this class, this isn't a nutrition class. It's not, designed to help you make a nutritional plan. But I think that a little bit of biological knowledge and a little bit of the prefix-suffix knowledge will help you in determining the health of foods based on the biological context. And so, what does saturation mean? Saturation means that you are completely, and I say you, the fatty acid is completely saturated with hydrogen. So when we talk about saturated, we're talking about being saturated with hydrogen. All of these little blue dots are representative of a hydrogen. Notice that every single white dot, which are the carbons, are completely bonded to 2 hydrogens each. Until you get to the end, obviously you need 3 hydrogens, but there are no open spots for the carbons because they're all saturated. They're full. They're all full of hydrogen. That is saturated. They are saturated with hydrogen. There are no double bonds. So what happens if you get a little bit of unsaturation, right? Notice that we have removed a hydrogen and we have removed a hydrogen. We've removed 2 hydrogens. So we have somewhat unsaturated our fatty acid. Well, because we have pulled off hydrogens, we need to bond these carbons to each other because they still need to have 4 valence electrons and they still need to bond 4 times, right? So now this carbon, instead of bonding to the hydrogen, now has to bond to this carbon twice. So it creates a double bond. This carbon has to bond to this carbon twice because it needs to bond 4 times. And so a double bond is created. This is an unsaturated fat, and this would be a monounsaturated fat, right? There's a single point of unsaturation. This is also a monounsaturated fat. You can have polyunsaturated fats, right? If you had multiple double bonds throughout, it just means that you would have fewer and fewer number of hydrogens. But as the molecule, or as the fatty acid loses more and more hydrogens, it becomes more and more unsaturated, meaning it becomes more and more healthy for you. Why is it healthier to eat unsaturated fats as opposed to saturated fats? And it has to do with the flexibility of the chain. When the fatty acid is completely saturated with hydrogens, it's very rigid, it's very locked in place, and it's therefore very solid at room temp. When you start pulling the hydrogens off and you start creating these little kinks or these little bends, all of a sudden the chain isn't so rigidly held in place, and it actually now kind of hinges from that double bond. And so the more unsaturated it is, the more double bonds throughout, meaning the more fluid it is, the more liquid it is at room temp. So a saturated fat is going to be very solid at room temp, and an unsaturated fat is going to be very liquid at room temp. What does that sound like? Well, your vegetable oils and your olive oils and your canola oils are all unsaturated fats, which is why when you put your olive oil in the pantry, it stays liquid at room temp. Whereas saturated fats are things like hamburger grease or other animal fat products, right? If you cook bacon or you cook hamburger, you're going to get animal fat that renders out of those proteins and renders out of that tissue. And so if you let it cool to room temp, you drain it off the meat and you let it cool, it's going to solidify at room temp. These saturated fats, because they are so rigid and so inflexible, they solidify at room temp. What does that mean? Well, body temp isn't that much warmer than room temp. And so if these animal fats are solidifying at room temp, they're probably going to partially solidify at body temp, meaning they can clog arteries, they can clog other small openings and ducts and stuff, and so it's less healthy than the unsaturated fats, which stay liquid and therefore stay fluid. Okay. The only thing I'm going to talk about in terms of trans and cis, that's more of an organic chemistry thing. Basically, trans is when you have the two hydrogens that flank the double bond are on opposite sides of the double bond. Notice that this one is basically on the north side, so to speak, and this one's on the south side. Whereas this one, the two hydrogens that flank the double bond are both on the same side. That's a cis, okay? That is a bent configuration. This obviously is a little more healthy in terms of the unsaturated fat. So you have trans fats and you have basically the cis unsaturated fats. Both of them are unsaturated, so they're both liquid at room temp. However, the trans fats are a little more rigid within the group of unsaturated fats than the cis configured unsaturated fats. So a lot of times you hear food products market that it's zero trans fats. That's obviously something to look for, because trans fats are more resemblant of animal fats, even though there is a double bond and even though they are unsaturated. Okay, so saturated fats are solid at room temp and are bad for the body, whereas unsaturated fats are liquid at room temp and are better for the body. I wouldn't necessarily say they're good for the body. They're still fats. Fat is good, or lipids are good for the body. You do need them, but you need them in moderation for sure. Okay, so the other class is the phospholipid, and the phospholipid is not an energy store, just like the triglycerides are. It is all about a biological membrane constructor, so to speak. They are made a little differently, but they still possess a lot of the same characteristics that the triglyceride does. Notice that this is very resemblant of the glycerol that the triglyceride head was made of. And so this is the glycerol. It's the same exact molecule that the triglyceride head is made of. This is a fatty acid, exactly the same saturated fatty acid. Notice that it's completely saturated with hydrogens. This one is showing you one cis-configured unsaturation, or point of unsaturation, a double bond, right? The hydrogens are on the same side, so this is a cis unsaturated fatty acid. That's why it bends, okay? So it remains fluid. And then the third tail, which in a normal triglyceride would be here, right, is now kind of bent around and is now here. And that third tail, which used to be here, kind of helps to make this phosphate group part of this phosphate head, right? It's still very much just like a triglyceride. It's just a triglyceride with two tails exposed, with the third tail kind of wrapped up in this phosphate to make this head. Okay, so what's unique about a phospholipid? When we talk about the triglyceride is a molecule that is completely hydrophobic. And what I mean by that is the triglyceride is completely nonpolar and therefore completely hydrophobic, meaning the entire triglyceride does not dissolve in water and it repels water, which is why when you put oil in water, it floats on top and it doesn't mix in. Okay, that's because they are nonpolar molecules and they are hydrophobic. They do not interact with water. Well, the phospholipid, on the other hand, has a polar head and nonpolar tails. So just like the triglyceride is nonpolar, the tails or the fatty acids of the phospholipid are nonpolar as well. But the difference between the triglyceride and the phospholipid is that the head of a triglyceride is also nonpolar, whereas the head of a phospholipid is polar. Why is that important? When the phospholipids orient themselves into this bilayer structure, the zone that is created is a hydrophobic zone, basically a zone of hydrophobicity, meaning no water exists in the tail portion or in the internal portion of this membrane. There obviously is water out here, and there's water on this side. Water can interact with the heads on this side. Water can interact on the heads on this side, which is really important because we know that cells often or need to live in an aqueous solution. They obviously have to interact with water, and so there's water in the environment, and there's also water inside the cell cytoplasm. So this would obviously be inside, this would be outside of a cell. Water is going to be found outside a cell, water is going to be found inside of a cell. So water has to orient and touch the boundary of the cell. However, water does not interact with this internal side because it's a zone of hydrophobicity. All right. We refer to these molecules being amphipathic, which is a cool designation for molecules that have both hydrophilic and hydrophobic parts. Okay. A phospholipid is amphipathic because it contains a hydrophilic head and hydrophobic tails. Okay, so hydrophilic is water-loving. It can interact with water. It's polar. The head is hydrophilic. The head is polar. The tails are hydrophobic. They repel water, which is why they stay in this zone of hydrophobicity. Okay, when we look at this diagram up here, this is a more detailed version of what the cell membrane looks like as opposed to this one. The cell membrane is a double layer of phospholipids. It has proteins embedded in them. It has cholesterol, which is also a lipid. And that's really important to note that because the cholesterol, just like the triglycerides, are completely hydrophobic and are therefore completely nonpolar, the cholesterol actually embeds itself into this zone of hydrophobicity because they do not interact with water. Again, if this is the outside of the cell, then obviously there would be water in the environment and it would be interacting with the tops of these phospholipid heads. We also would have water on the inside of a cell interacting with the underneath side of this membrane, but there are no water molecules in. Water can actually travel through protein channels called aquaporins, where they can move through these tunnels without actually interacting and touching these nonpolar hydrophobic tails. Okay, so that's it for the membrane. We will have another more in-depth, detailed version of the membrane in a future video. But for now, two different kind of versions of lipids, both phospholipids and the triglycerides, used in entirely different ways, but are both definitely lipids. So if that's it, bring your questions to class. See you later.